Hello guys, welcome to my channel. So as you know, we are studying about the protein function. In the last video, we discussed about the carbon monoxide. So now there is one more thing. Before we move further in the protein function, we'll discuss about that. So that is sickle cell anemia. Sickle cell anemia, right? So this is a molecular disease of hemoglobin. Hemoglobin. Right, so we'll not discuss on the medicine level, but we'll just discuss on the molecular level as we are studying about the hemoglobin and the binding of oxygen. So as the hereditary human disease sickle cell anemia demonstrates strikingly importance of amino acid sequence in determining the secondary, tertiary and quaternary structures of globular proteins and thus their biological functions. This because of this sickle cell anemia, the importance that we studied all about the primary, secondary, tertiary, quaternary structures of this protein. Why? Because this sickle cell anemia, so why this is really important. So almost there are like 500 genetic variants of hemoglobin. How many? Like there is like 500 genetic variants of hemoglobin. So these 500 genetic variants of hemoglobins are known to occur in human population, but there are only few that are quite rare. So most variation consists of difference in the single amino acid residue. It's just a single amino acid residue. So the effects on hemoglobin structure and function are often minor but can sometimes be extraordinary. Usually just because of that single amino acid residue, the structure and function will be just a minor change, but there might be a sometime extraordinary change as well. So each hemoglobin variation is a product of an altered gene. Why? So I will write it down here. Each hemoglobin variation is a product of altered gene, right? And the variant genes or the altered genes, you can say, right? The variant genes are called alleles, right? So because humans generally have two copies, right? Humans generally have two copies of each gene, right? Two copies of each gene, correct? An individual may have two copies of one allele, right? Just two copies of one allele, that is called homozygous for that gene, or one copy of each of two different alleles, that is called heterozygous. We'll study that in deep, but just for this sickle cell anemia, just understand that, right? All right, so sickle cell anemia occurs in individuals who inherit the alleles for sickle cell hemoglobin from both the parents, right? So the erythrocytes of these individuals, those have sickle cell, are fewer and also abnormal, right? So the erythrocytes will be fewer and also abnormal. In addition to an unusually large number of immature cells, the blood contains many long, thin, sickle-shaped erythrocytes. Right, so as you can see on your screen, the picture on the left-hand side, you can see that's the normal hemoglobin, or rather normal erythrocyte picture, and on the right-hand side, that is the sickle cell erythrocytes that you can see, right? So it has a different kinds of shape, spiny or like sickle shaped. So when hemoglobin from sickle cells, the sickle cells, it's so what's the hemoglobin is called of sickle cell? Okay, sickle cells, and hemoglobin is known as hemoglobin S, or also known as HbS. right? So when hemoglobin from the sickle cells, also called as this HbS, is deoxygenated, remember deoxygenated, right? Is deoxygenated, it becomes insoluble and forms polymer that aggregate into tubular fibers. Alright, so I'm writing this down here. Okay, so when from sickle cell, that is this HbS, right, is deoxygenated, it becomes insoluble and it's insoluble and forms what it forms. It forms polymers that aggregate into tubular fibers. How? So we'll see that. So the normal hemoglobin, that is also HbA, remains soluble on deoxygenation. So what happens to this HbS? If it deoxygenated, it becomes insoluble, but for the normal hemoglobin, it remains soluble. So that's the reason the insoluble fibers of this deoxygenated HbS causes deformed sickle shape of the erythrocytes, and the proportion of sickle cells increases greatly as blood is deoxygenated. Alright, so I will show you with the diagram. Alright, so you can see this, right? So for example, I'm just drawing, for example, this is alpha 2, alpha 1, beta 2 and beta 1. So this is the HbA, that's the normal hemoglobin, for example, and here let's draw this. So what will be the difference? So this is the same as alpha 2, alpha 1, beta 2, beta 1. So this is HbS. So this is the sickle cell hemoglobin. And what happens in this? So in this too, the altered property of this hemoglobin that has sickle cells result from a single amino acid substitution. So which amino acid is it? It's instead of VAL, that's valine, instead of GLU, glutamic acid residue at position 6. At position 6 in the 2 beta chains, right? In 2 beta chains. So what happens? So this is 2 beta chains in this HbA and this is 2 beta chains in HbS. So in this sickle cell hemoglobin, so the substitutions from valine instead of glutamic residue at position 6 in this 2 beta chains. So the R group of the valine, right? So valine has the R group. So R group of the valine has no electric charge, but whereas this glutamate has a negative charge at pH 7.4. So this glutamate has negative electric charge at pH 7.4, right? So the R group of the valine, this has no electric charge, right? No electric charge, and glutamic acid has negative electric charge at pH 7.4, right? So hemoglobin S therefore has 2 fewer negative charges than the HbA because glutamic acid has been replaced by valine, right? So this hemoglobin S has what it has. So the hemoglobin S has a 2 fewer negative charges than this HbA. How? Because it is replaced by valine instead of this glutamic acid. As the replacement of this glutamic acid, this has negative charge and this has no electric charge. That's why it has a 2 fewer electric charges. Why it has 2? Because it has 2 beta chains, right? 1 and 2. That's why it has 2 fewer negative charges than this HbA. So the replacement of this glutamic residue by this valine creates a sticky hydrophobic contact position point at a position 6 of this beta chain, which is on the outer surface of the molecule. Right, so what happens? So for example, I will just make this picture here just as a single circle. If this is like there is an interaction between these molecules, right? Interaction between these molecules. So what it will do, it will create a like a strand formation. This will be like something like this. This will be like strand formation, and from this strand formation, if it goes on, like it will become like more and more sticky. So it will become like really like this will become like an alignment and crystallization, or you can say as a fiber formation. It's because of this glutamic residue that is replaced by this valine. So how it's done? Because this replacement of this glutamic residue by this valine, what it does, it creates a sticky hydrophobic. It will create a sticky hydrophobic, right? Hydrophobic contact at the position 6 where it is replaced at the beta chain, which is, and this is on the outer surface of the molecule. So this sticky spots, what it does, causes this deoxy hemoglobin sickle cell molecules to associate abnormally like this with each other, forming the long and the fibrous aggregates characteristics of this disorder. Right, and sickle cell anemia is life-threatening and painful. Why? Because people with this disease suffer repeated crises brought on by physical exertion, also known as sickle cell crisis. They become weak, dizzy, short of breath, and they also experience heart murmurs and increased pulse rate. So the hemoglobin content of their blood is only about half the normal value of 15 to 16 gram per 100 ml. Because the sickle cells are very fragile and rupture easily, that is why it results in anemia, right? That's the lack of blood. Why? Because the hemoglobin or the sickle cells are very fragile and rupture really easily. That's why the normal value is like almost like half of from the regular values, right? And even more serious consequence is that the capillaries become blocked by this long abnormally shaped cells, causing severe pain and interfering with the normal organ functions, a major factor in the early death of many people with this disease, right? Because it's like sickle cells and different kinds of shape, the erythrocyte, it might, like capillaries are really small, so it might block the capillaries, causing severe pain, and like it might, if it blocks the like capillaries going through any important organs, like it can cause a death as well. So without medical treatment, people with sickle cell anemia usually die in childhood. Curiously, the frequency of sickle cell allele in population is unusually high in certain parts of Africa. Why? Because investigation has been done into this matter and led to finding that when the heterozygous, the allele conforms a small but significant resistance to lethal forms of malaria. Right? So the heterozygous individuals experience a milder condition called sickle cell trait. Only about 1% of their erythrocytes become sickle on deoxygenation. So these individuals may live completely normal lives if they avoid any vigorous exercise or other stresses on the circulatory system. And the natural selection has resulted in an allele population that balances the deleterious effect of the homozygous condition against the resistance to malaria afforded by the heterozygous condition. Alright guys, so this was sickle cell anemia on a molecular level, not on the medicine. So we'll definitely study sickle cell anemia again when we'll study the system and when we'll discuss about the disease. But when we are studying about the hemoglobin and the protein structure especially, so that is why it was just important to know how this sickle cell anemia affects when the hemoglobin structure has been changed. Alright guys, thank you guys for watching, and don't forget to subscribe my channel. Thank you. We'll see you in the next video. Thank you.